now have your modeler in place, executable and working, and I hope that you've actually gone through the tutorial yourself. So once again, just to provide a little bit of context, we will try and go through this tutorial process. So we are at the modeler tutorial link, which is here. And there are all kinds of interesting modeling procedures if you make this part of your research. But that's not what we are interested in right now. We're just interested in learning how to do the basic modeling as an important exercise. So this is modeling. Now, when you downloaded your all the files that are required, you'll see that the files that are important in the basic modeler process is what is called the profile build.py, the compare.py, the align2d file, and the model single file. And all the PDB files that you would require are right here. Okay, and also the intermediate files that you would require are right here. Now, because the Python that I have is 32-bit, the modeler program that I installed was also 32-bit, and I think this is crucial. There seems to be some incompatibility between the Python and the latest version of modeler, and we're trying to get away from these incompatibilities. Even right now, when I try to run it on my work desktop, which is where I'm working right now, I immediately got the log verbose problem. So what I did immediately was I went and I checked my, I typed in idle, and you'll see that it said 32-bit. Okay, and the moment I realized that my idle was 32-bit and I'm using an older version of Python than you are, I immediately went and I downloaded modeler, and the whole process, the log verbose file, got fixed in almost instantly. Okay, and I was able to run my first code just to make sure. So one of the things that you might want to think about is ensure that there is compatibility. If you need, just for the time being, to deinstall and go to your 32-bit, and I think this is one of the issues, is that for Mac, as one of the students, I think Benjamin mentioned, for the Mac, the 32-bit is the version that they have, and it's possible that your Python is 64-bit. So if you go to the 32-bit Python, the compatibility will be dealt with. Again, I did get the log verbose file error now, but the moment I downloaded and instantly installed a 32-bit version, that problem went away. Okay, all right. So these are the files. These are the files that we have. Now let's go to the basic modeling tutorial. Okay, and so the basic modeling tutorial is basically, as it happens with any tutorial exercise, is you find a sequence whose structure you don't know. Okay, and then what you want to do is you want to find a template that whose structure is known and that bears either a sequence homology, a functional homology, despite the way we define homologies, sequence identity, sequence similarity. So this is what gives you that confidence. If I know a structure whose sequence and whose function I know, and if I find a sequence of an unknown of whose structure I don't know, but has identity or similarity with my template sequence, then I can confidently believe that that the sequence of the unknown structure is likely to have a similar structure. That's the foundations on which this notion of homology modeling and I think modeler is the, I would say, the standard bearer for this kind of is a standard bearer for this kind of modeling. Okay, so let's look at the files that we're looking for. So you've presumably gone and installed, downloaded the zip format for Windows, or I suppose for Mac, and those are the files that I showed you before. So the novel gene for lactate dehydrogenase was identified from the genomic sequence of the Trichomonas vaginalis, TVLDH. So now let's go to our folder, and you'll see that TVLDH, all of these are the files whose sequence we didn't know. Okay, and then so these TVLDH. So again, remember, we hypothesized that TVLDH arose from TVMDH by convergent evolution. So comparative models were constructed to study the sequences of structural context. Once again, what they're trying to say is we have a sequence whose structure is known, we have a sequence whose structure is not known. As soon as we find similarities in terms of their sequence, and we can, from a homology standpoint, go back in the evolutionary period and think in terms of, well, these functions have also evolved to be similar, that means it's likely that the structure is also similar. Okay, and so let's go and look at the sequences that we have. So one is what is called the alignment file. Okay, and this is the alignment file, and this is in the PIR format, the PIR format. So again, you were provided all the files that you need, and you'll see that there is a similarity between this and the FASTA format. Now, so once again, the sequence that's known, it's P1 is given as the, and here's the designation TVLDH, that's the sequence that's known. You see the caret sign, the greater than sign, which tells you that that is the beginning of a sequence. Then in addition, the PIR, the Protein Information Resource file, also has a, and look, they say here, the PIR format readable by modeler. And so you see that here's a sequence that of an unknown, of the sequence whose structure you want to find. Remember, it ends in an asterisk, so watch out for this asterisk. And in addition, there's one difference between the PIR format and the FASTA format, and that is after the designation of the protein identity, you have the designation of additional information. Now, here you'll see that these are actually places between these colons. Information actually has to be filled, but that information is mostly structural information. We don't need any of that information right now because we don't have any of the structural information, but you still want to make sure that the format is correct, is exactly the same. Okay, and then once we create the PIR formatted files for the templates whose structures we know, we'll see how those differ, and we'll see, because those come from real structures, we'll see that this information between the colons is actually filled. Okay, so this information you can read. It tells you more about what I just said. Now let's look for the file. Remember, we're looking for the tvldh.ali file, and so let's look into our code. And you'll see the tvldh.ali file, and I'm going to open it with Notepad, and you'll see that's exactly the sequence that is available to you. Okay, so that file is actually available. Now, what we're trying to do is we're going to run this build profile.py, the Python, it's a Python code. And this is, build profile, profile build, it's exactly the same thing. Now, because, once again, let's look at my folder, and you'll see that my folder has here the location of where my information is. Okay, and my modeler file, my modeler executables are sitting in program files 86. Okay, so there's my modular 9.24. Okay, and if you double click on it, you'll have all these including the examples, but I'm not going to worry about it right now because the files, so the executable is here, but the files that I have created are in this particular folder. So it's this PC and the D drive. So what I do, and typically this is something that you might also want to do, is once you have a PC, if you have a desktop and like me, you're doing a lot of research. What you want to do is you want to say, for example, this is a 2 terabyte hard drive. So a 2 terabyte hard drive, I've designated, for example, if you look at this PC, you'll see that I've actually designated about 500 gigabytes to my local disk C, and about the rest of the the rest of the terabytes. So this is 2.5 terabytes. So I've designated the rest of this to my drive D. So what you do is you put your executables, you put everything that you've downloaded into C, and then all your data files you want to store in D. So this way what happens is if you partition it and if you have trouble, you can say, well, I don't need any of my files for C because I can always download and install them. But the data is what is really crucial. Okay, so again, we're back here. We've looked at our TVLDH alignment file. And now we're trying to run our modular first file, that is the profile build. It also shows up as build profile. This is a relatively older file. And this is Python 3.6. I think we're right now at 3.8. So what I've done for each of these files is I have put the full pathway that I have, right? And where is the pathway? This is the path. Okay, this is the path. This PC, I'm looking at the D drive. I'm looking at a folder called Bioinformatics Education, where all my data of all my coursework is BTech 5333-2020, which is this file, and class 9. Okay, so it's likely, so I need to change this to 2020. Okay, so all of these, so every file that I'm reading is here. Okay, and that tells you that okay, and these are all the files, which means I want all the output files to be written to this folder. Okay, so I've created this file, and you'll see that we have the PIR file that was already there, PIR file. Remember that PDB95 file, which was the downloadable file that we had to separately download, that is also sitting here. And again, so everything is being pointed to the right place. The TVLDH file is right here. And the files that I'm going to generate out of this, including the log files, are going to be stored here. Okay, so everything is copied here and everything is being stored here. Now, you'll see that these versions of these files are already there, and while you run your code, I think what they've done is they've not only provided the input files, they've also provided the output files. Okay, so we do our usual thing with the Python program, and we are going to run, for example, there's code. And so we're going to do run Python shell, run module F5. I saved it because I recently changed it, and you can see this running. And now obviously it's going to take a few minutes. This is a relatively fast computer, though it's more than 5 years old, so it's not going to run at peak performance. But you can see this going. Now you can look at exactly all the information that you're seeing in here, but it's kind of difficult. There's a lot of things that are a bit of a black box. But again, So what files get created? Now let's go back to our tutorial. So we begin to have an understanding of the files that we have. Okay, so build profile, and once again let's begin to, we've already discussed this when we had our class. So non-redundant PDB sequence is a 95% sequence identity into this database. Okay, so that's the PIR file. Now we can look at the PIR file, and what is the PIR file? It literally has all the, and we can open it in Edit Notepad, and you'll see it has these PIR formatted files of all the structures in the PDB. Okay, and all the structures in the PDB, but not every structure. Remember, we want to make sure that things that are too similar don't mess up our results. If something is too similar, oh, look at all these structures, they're too close to each other. So for example, if you are ever in this field, you'll realize that lysozyme crystallizes, forms these beautiful crystals very quickly. So if you wanted to buy a new instrument, you wanted to test it out, you will make lysozyme crystals. So obviously you don't want all your thing to be burdened by lysozyme, because if there's a single hit, you'll get 300 different hits from lysozyme. So you want some dissimilarity, so 95% similarity. And so while so this is the PIR file of, like, I would say about 40,000 different structures, the PIR files. And so when you have the comparisons, this is what the comparison is. And so remember your notes on, or your thoughts, or whatever we learned on binary alignments, so sequence versus sequence. So the sequence of interest, the TVLDH, is actually comparing with each of these. That's why we need this PIR file. Now the bin file has the structural information. So the bin file has the structural information, but it's not, but it's a binary file, so it's not going to be openable in into anything useful. Okay, so but again, the bin file is required eventually. So we've run our first code. Okay, and we've run our first code, and so for example, what it's done is it's created a build profile PRF file. So what it's done is it's gone ahead and done an alignment. It's gone an alignment with every sequence in the PIR file, and once you put every sequence in the PIR file, it's gone and done some alignment, and now it's created a PRF file. So let's go take a look at the profile build.prf file. Okay, and we can see this PRF file. So what it's done is it's taken the TVLDH, the sequence whose structure we want to solve, and it's gone ahead and it's aligned again. So remember how the gaps, etc., have been formed. Okay, it's gone and aligned it with different things, and it's gotten 36 different alignments. Now we have to kind of take those alignments and do something else with it. One more thing about the PIR file. So you'll see, for example, remember how in the TVLDH alignment file, all of these things were empty. The same PIR formatted file will have more information. So you see all this information, but so this is the structure. So that was sequence, because that's the unknown. This is structure, so structure X. This tells you the PDB ID. So 1 UUUQ, and this tells you, remember how I told you the colon, the space between the colons. In this case, we don't have structural information, but you'll see a lot of structural information that's already built into this. So this tells you it comes, so the PIR format will actually have information related to the crystal structure. 431 amino acids, for example. It'll tell you it's the A chain. It'll tell you the name of this particular enzyme, that is chain A. So it tells you, what the scientific name of this organism is, Salvibrio mixtus. I don't know what that is, and it says that the common name is Salvibri, etc. So you have all this information. So all that information between the colons is actually completed. So this profile build.prf, it went and it said, out of those 40,000 or 35,000 odd sequences, here are the best results that we have found. Now, moving on. Okay, so it's done all of that alignment. So there's something else that we were supposed to look at, and that is so this is the alignment file. Okay, this is the build profile file that we just ran, and what it ended up doing was it created a build profile log. So what's in the build profile log? Because you want to be able to track the errors and all of these instances of information kind of run through really quickly, you want to go ahead and check and see, let's say you wanted to track some of this information. All of this information that you saw here is actually stored in the build profile log file. Okay, so and you'll see it's exactly the same. It's exactly the same. In case you wanted to track something, but looking at it on this Python executed results screen is a little too complicated, you have all of this stored in a log file. Okay, what else? We looked at the PRF file. Now let's look at the build profile alignment file. Okay, so this is exactly like the PRF file, except you have very dedicated ways of determining which things in the PRF file, which proteins in the PRF file whose structures were known, we actually find some alignment. Now this is obviously a lot of information, and the next steps will actually winnow it down to something a little more useful. Okay, so if you look at the profile build, we look at the profile build log. Now let's look at the profile build.prf. So if you look at the profile build.prf file, you'll see now they have a lot more information than was created in the original thing that they have created here. And that's why it was a little, it took me a while to figure out what exactly was going on. So if you really look at this line, for example, this line, for example, all these are the lines that are represented here. Okay, but now this the newer version of Modeler gives you this additional information. It actually shows you this. So this is our unknown, or a sequence whose structure we want to find, and this then tells you, for example, how these alignments take place. Okay, now what else do we need to know? So let's look at this. It says what's more, what columns are really important. So for example, it tells you there's 335 residues. It goes from 1 to 335, and then it says, well, how many of these? So for example, in this second protein 1A5Z, you can see 1A5Z, you'll see that there are 312 amino acids in here, of which residue from 75 to 242 match. Okay, and so there's a whole bunch of additional information here. So what do they say? What's the most important thing that you need to know here? So the first 6 commented lines indicate this information. Okay, that is the information about the TVLDH. Now in terms of what is the most important, where does it say? It says the most important columns are the second, which is, and again let's go up a little bit, is the second is the name of the PDB file from which it comes out, the 10th, so second, third, fourth, fifth, sixth, 7th, 8th, 9th, 10th, okay, the 10th, 11th, and 12th. So what is the 10th column? The second column reports the PDB code, okay, from what PDB that particular structure was found. The 10th column, the 11th column says that 33% of the residues in this sequence have an identity with the TVLDH, 33%, and the 10th column tells you how many of those residues are identical, sequence identity. So the 10th, 11th, and 12th. And what's the 12th? If you remember our blast search gave us an E value, an expectation value. And just to reiterate what expectation value is, it is the probability that that particular match happens purely at random. So once again, if you look at this number, 0.32 E-05 tells you it's 0.32 times 10 to the negative 5, which means the probability of that match or these identities aligned is virtually very small. Okay, so these are the alignments we find. So what we're going to do is we're going to say, well, yeah, these are small, but we'll go with the 5. We'll choose the ones for which the randomness is 0. Okay, so these are the ones with the 0 are our top scoring. And then what we do is we go to what is called, then we find the ones which are 0. So for example, 5 MDHA, 7 MDHA, 1 SMKA, 1 CIVA, then the B8PA and the BDMA. We take those 6 structures or 7 structures and we say those are the ones, or the 6 structures, and we say those are the ones which have a 0 probability of the alignment being because of random chance. And then we look at our compare.py. That's the next code we're going to use. Okay, so I'm going to remove those and then look for the compare.py. Open with idle. And so once again, make sure that I have I'm in the right place. Okay, that is 2020. Okay, now the B8P, BDM, CIV, 5MDH, 7MDH, SMK are PDB files that are already there. Okay, so now I already have my sequence comparison. That was the build profile process at that. Now I'm actually going to look at these PDBs and then go into this binary file where the structures are present and begin to do an alignment that is structural. Okay, so I'm actually now going to take that and I'm actually going to align them in a structural way, and now I'm going to incorporate all my PDB files. So to see the most appropriate template, okay, that's the thing. Now we're going to look at the structures and we're going to find which of these templates are the best suitable. So now it's almost like the build profile process did something that was kind of a rough, because we went over several thousand PDB structures, the sequences, and did an alignment. Now we're going to take the ones which have the highest results, and we're going to do a more ingrained comparison. Okay, so now what we're going to do is we're actually going to go in and do a more ingrained analysis. Okay, so now you want to see which of these templates are the most ideal. So let's look at our compare files. So you have all the PDB information that is in here. And now you look at this. For example, we're going to do aligning of the sequences, and then we're going to do alignment of the structures. And once again, I have make sure that we are in the right place. Okay, and this is where all this information is located, and we're going to create these files. So this is compare. So now we're taking only the top 6, the ones that actually had 0 in terms of E values, and we're going to run these in what is called the compare.py. Okay, so now I'm going to run it. I'm going to save it, run it, and of course it's going to do a comparison, and it's going to give me something a little more definitive. And what it did, what you see here, it gave at the end was it gave me something that was like a phylogenetic analysis. But now what it did was it began to incorporate the structural information also. Okay? Now, let's look at how do we interpret this. So what you see here is kind of an issue. And that issue is what happens, for example, when you put your example folder elsewhere. So it's actually giving me this entire pathway, which is not really very helpful to me. Okay, so now let's look at, for example, there's not much we can do in terms of this, but let's say, for example, we look at these files. So B8PA is here. B8PA, it's the exact same alignment, but except one of them is missed out here. Okay, now what do we see here? These are our alignments. That's the results we see. But obviously, because I use this full path, it's treating the whole path as the name, and obviously it's not formatted to take that whole path into account. But one of the things that we could do is, so for example, we can use this to compare. Again, one of the things that you might want to do is put your example folder in the modular folder. In this case, you can do the executing right from there without having to worry about issues like this. And this happens just once. So again, we'll see that this is the alignment. It created a thing like a phylogenetic tree. So this will tell you exactly, how do you determine what the template should be. Okay, so now let's look at the reasoning that they use for what the template should be. So it shows that CIV and 7MD are very close. See that? In a phylogenetic analysis, they come really close, both sequentially and structurally. So remember, it's not just a sequence comparison, it's also a structural comparison. But 2.4, which is a better resolution, so hark back to your to the resolution, the definition of resolution we've studied. Go look at the video if necessary. Now, so obviously you would say 7MDH is the winner compared to 1C1. So we knock out that guy. Okay, so the second group of structures, 5MDH, 1BDMA, and 1B8PA, share some similarity. 5MDH has the poorest resolution, so we only take the 1.9 and 1.8. In terms of resolution, remember, the smaller the number, the better it is. So 1SMK is the most diverse structure of all the possible templates, but it has the lowest sequence identity. Okay, so we're not really looking at that too, because the sequence identity is really poor. So then we say the crystallographic R factor, which is 16.5 or 16.9, is the best. And what is R factor? R factor, simply put, is a comparison between the density that you get in terms of the data from an X-ray machine and the model that you build around that density. Okay, so when you pick 1BDMA, okay, because it has the best resolution, it has the best sequence identity, 45% or 50.5, okay, and so you pick this sequence. So now this is our sequence of choice. So this is the template. We know the structure. The 1BDM is a crystal structure, and what we do is what is called the align 2D. So now this is actually the very tricky part, because remember, it does not have 100% identity. The identity is not very good, but it's better than the rest, and its structural characteristics are also very good in terms of the better R factor, the better resolution, etc. And so we want to now align it. And this alignment is really important because remember, in a homology modeling, we are taking an unknown structure and we are building it over the scaffold of a known structure. So for example, you want to say, oh, there's an alanine there, which means my alanine, if it sequentially matches, that would be the right place. So that would have a similar structure. And this is therefore very important. So then we're looking at the align 2D.py. Okay. And again, this is where I discuss homology modeling to be not just a science, but an art. Now, this tutorial is pretty stable. It's really well developed. So you don't have to worry too much about there might be some mistakes or something. But don't rely on the software to just give you that information and just put it into the next step. That's not what we want to do. What we want to do is make sure that we use our knowledge of some of the biochemistry of this protein, which will help us determine how to align and put in gaps and things like that. Okay, so that's our knowledge. So if you were doing this as a research project and you really understood this protein well, you would take a step back and say, great, align gives me a rough sort of an alignment. Now I've got to take in my own knowledge and create gaps, etc. Okay, so let's run align 2D. So again, this is the code. So you know that you've chosen the 1BDMA and it comes from 1BDM.pdb, and the TVLDH is a file that you need because eventually you need an alignment of both. Let's go into our files and look at, delete the rest, close these windows and look at the align 2D Python code, which is exactly how it is. And once again, I have actually included all of this here. The same thing. I've just changed this a little bit, so I know I'm in the right folder. And it tells me where I want to create my alignment files. I want to create something called as the PAP file, and we'll see what that format is. Here's the alignment file that it uses from our first build profile. It says here's the BDM PDB. It's the chain 1BDMA. It comes from 1BDM.pdb. We can expand this a little bit to see that from start to end we are still invoking chain A. Okay, and that's really important because if this is a heterodimer of some kind, or it has subunits A and B, and it's the A and B are not equal to each other, it's important that we specify what that A is. If, for example, it doesn't have subunits, it doesn't have a chain A, B, etc., you just leave this region blank. Okay, I'm going to save it and I'm going to run it. And once again, we are going to do an alignment, and what's going to come out of this is the alignment file. So we're going to have to wait for a little while, because remember, this is also a structural alignment, and this is really very important, because if this alignment is incorrect, and that happens quite a bit because the software does the best it's going to do, if this alignment ends up being incorrect, you're going to find that your resulting structure of the unknown is going to be wrong too. Okay, and that's very, very important. So let's look at our result, and it'll tell you that you have the PAP file, which is again like a binary alignment file. So let's go in there, take a look at our TVLDH 1BMA PAP file. And the PAP file is like an alignment file, right? So for example, it's doing its alignment, and the alignment looks like it's pretty good. So for example, if this is M, K and S, yes, it's going to make these adjustments. That's what the modular software does, because things are never going to be equal. Wherever you see an asterisk, it means there's an identity. Otherwise, it's a similarity. Now, obviously, here, for example, there is a good QV, Okay, there's a QV identity between glutamine and valine, but here there's a pretty big gap. So now it's going to be a challenge for this software to make sure that it somehow rationalizes the structure in this area and rationalizes the structure in this area. So that's tricky. The rest of it is not too bad. Now, typically, if you already know what your structure is going to be, or what your structure is, your template structure, and you know what your sequence is going to be, then you don't do the first two steps because you already know your template. Then this align 2D becomes really crucial because this gives you some sort of an alignment. And then you go in there and you put gaps in based on your best guess, based on your knowledge of the biochemistry of it. You take it, you put in gaps, you say, okay, well, this is not correct. A gap belongs somewhere else, okay? This is then up to you. So if you were to do this as a research project and you know your template, you would start at the align 2D because you had already decided what your structural template is, and you already know obviously the sequence is what you want to find out. So this is really very important. Now, the PAP file tells you about the sequence alignment, but the ALI file, the TVLDH align file, that is the crucial, because that then creates the alignment in the peer format. So you see 1dBMA, this is all this information. So this is molecule is malate dehydrogenase, chain A, tells you it's an enzyme, and that's the enzyme consortium nomenclature, which will tell you exactly what that enzyme is in terms of its category and subcategory, and eventually the identity of the protein of that particular enzyme. Here's some additional information related to the structure. Now, this alignment file is really important, and most people begin to struggle when it comes to this kind of alignment. And the reason why people struggle is because, remember, so for example, no matter how many gaps you have, you see these asterisks, it has to be exactly the same. Okay, exactly the same. So all this is really important. So for example, here's something else that the modeler software is going to have to rationalize. So again, very often people, this is one off, and then it gives you an error, and then you don't know what to do. This is very, very crucial. Okay, very, very crucial that these are perfectly aligned. And even though, for example, this is a sequence that we don't know, okay, whose structure we want to find, we have to make sure that even no matter what you do, the number of things you have in here, including gaps, is exactly identical. Okay, now I'll just give you another thing. What happens, for example, if you have, well, one protein whose structure is known would really model this part well, and another one will model this part well. And let's say then you have two different templates, and that's fine. You could have up to 100, you could have an unlimited, well, I don't know about unlimited, but you could have many. So then what you would do is you would say, I want to put in this, okay, and which means you're just going to put the first part because that's the one you want to template with this part, right? Then you put another C structure like this, and that would be the second part, which would align with this thing. But remember, every structure that you input, even if you just have amino acids up to here, you're going to have dashes here such that these asterisks match. Very, very, very crucial. Okay, now the align 2D builds this, okay, builds this for you, but you would still, if for things that you don't know about, you're going to have to go and put in your own gaps, and that's really crucial. Okay, that's as far as the alignment is concerned, and that's all the information we actually have. So I'm going to delete these two files, and now let's look at our model single.py. That's the last thing in the basic tutorial. And so run again. The SOAP is obviously is outdated. It was kind of an information acquisition and transfer protocol that was developed, which is not being used anymore. Once again, I need to update this to the folder we are looking at. It tells, so let's look at this file. It says this is the alignment file. This is crucial, right? That's the file that had this kind of alignment, this alignment file. Okay, that tells you that's the alignment. That's all the information that's already in there. And then it says the knowns are BDMA, the sequence is TVLDH. Now if you have more than one knowns, you're just going to have to add them here. Okay, and it says, don't worry about this thing. And so we're going to go from starting model 1 to starting model 5. Now the model is based on some internal metrics of internal energy metrics. Now the internal energy metrics is something that we really don't have a lot of control over. So what it means is you're going to have several versions of the structure. Now just because the software decides that something is the best structure doesn't necessarily mean it is the best structure as far as your research ideations are concerned, right? Now, but because it takes a long time, I'm just going to go from model 1 to ending model, from starting model 1 to ending model 2. Okay, and that's what I'm going to look at. So I'm just going to get model 1, model 2. We'll just look at model 1, that assuming that's the one that's the best, but we can go up to 100 if you're not really sure. But again, it takes a long time, so that's something to think about too. Okay, so what I'm going to do is, and this is the last part of the modeler tutorial, so I'm going to let it run. And as it runs, you can see that the new information is already being built. So it looks like it's done. Okay, this DOPE score is some sort of an energetic score. The GA341 is another kind of score, and you'll see that it has saved the information as B99901. So these are intermediate files. We can see what they are, and that's kind of these alignment, these continuous comparisons of of each amino acid is built. All these are some parameters in terms of energy and position and things like that. That's also being built at the same time. So these are files related to that. Again, I don't know what an RSR file is, but this is a file that we're really interested in. And so, for example, I'm just going to, you can use Python or anything you want. But I'm just going to use a code called Rasmol, which is simple to use. It doesn't have all the functionality, but so for example, that's the TVLDH that we have built, and that's a sequence that from the starting sequence using the template. Okay, and so for example, you can display in different ways. You can display wireframe, obviously. You can display space fill, display sticks, and you can use rotate, and I think this is used to translate. This is used to translate. The right button, the left button is used to rotate, and the middle button is, I thought it was used to zoom in and out, but I don't know. Now Rasmol has some other interesting things. So it has a command line thing where, and I think I've forgotten the commands, where you can choose. So let's say, for example, if I color everything monochrome and it's white, and then if I select, I'm just going to select, and again, I know very little in terms of remembering these commands. If I say select 45, it's going to select 5 atoms. But now only the 5 atoms are selected, right? Now I don't know what those atoms are. It could be, and then I'm going to say display as a ball and stick. You see the ball and stick? That's displayed as ball and stick here. Oops, where did it go? Well, what I'm going to do is I can color red, and now you know exactly. So you can actually manipulate each of those. And for example, if I want to change that display to space fill, I can look at the space fill of just the thing that I'm interested in. You can click on this and it'll tell you what residue it is. So the one which I randomly chose, it's an alanine, obviously the five atoms would be. And now if you want to go back to, you can then select something else. Select 69, 68, and I think you can do 68 to 84, and tells you all the residues. And so now anything that you do is going to change. Okay, and then I can say, well, I'm going to put my shapely, that tells you the color, but I can display it as backbone. I can display it as ribbons, and it'll tell you what, looks like it belongs to some kind of a strand. I can change the display. So again, whatever happens only goes to those things. Now if you say, well, I want to go back to everything to how it was, I can say select all, and then if I say space fill, and again, whatever I'm typing here, that information is also available, that I can type here. You can also type it in the command line, but I can't remember all of them. So anyway, this is our structure. It's pretty straightforward, but you have to be very, 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 very careful. You're going to get information that you're going to get errors. Most of the errors are if, for example, your input files are not in the right place, if your alignment files are not properly aligned. So that those asterisks don't meet. So for example, if you add gap but you forget to compensate in another sequence and your asterisks don't match, you're going to get errors. So it always pays to begin early, to start work on this really early, and go from there. Okay, yeah, I think that's about as much in terms of the tutorial of this particular software, and I think I wish you good luck. And the next tutorial I'll create as soon as I can will be on the docking. Okay, all right, I'll sign off now.